All right, let's take a closer look at the second process of the central dogma. This is changing the DNA code into RNA so that it can be made into a protein in the cytoplasm. This process is called transcription. This first step of the process is very similar to DNA replication. In transcription, the double strands of DNA are opened up and unwound around the site of the gene. This process is carried out by the enzyme DNA helicase. This will expose the two strands, one of which tells you how to make the protein, the coding strand, and the other is the non-coding sequence, which will be complementary to the coding strand. The second step of transcription is to actually transcribe. The enzyme RNA polymerase, which is different than DNA polymerase because it adds RNA bases and builds an RNA polymer, builds a complementary strand of RNA which will become the messenger RNA after further processing. The next step is for the transcript, which contains both exons and introns, to be spliced. This will attach the adjacent exons together, these code for the protein, and remove any introns, which are non-coding sequences. The final processing step is to add a cap at the front of the messenger RNA. This serves as a start signal for translation. At the same time, many A bases are added to the other end of the messenger RNA, known as a poly-A tail. The length of these A's seems to be predictive of how long this message will remain in the cell, and therefore how much of the protein will be made. In the final step of the process, the mature messenger RNA is exported out to the cytoplasm. In the cytoplasm, the next important step of the central dogma is carried out: translation.